Hello, everyone. I'm a new assistant professor in the Department of Pathology and also an affiliate member in Bioinformatics and Computational Medicine, as well as Biomedical Engineering. So our lab is located in the second floor of the BSRB building. Um, we're a lab focusing on spatial system biology for looking at all of the type of RNA, protein, and epigenome biology with spatial coordinations. The topic I'm going to present today is on spatial proteomics. So as you could imagine, and I apologize for people who have seen this slide before, this is my favorite metaphor to start with the talk, is that this is a map of the median housing price by zip code in the United States. As you could appreciate here, the median housing price are not just based on the actual build of the house, but the price is majority based on the location and the neighbors. It's the same thing as our tissue. So our cells are not independent. They actually are organized and are dependent on each other to form as a community. The region we are specialized have our interest in is the human hippocampus, as this is the center of the limbic system, which are regulating the memory, emotion, learning, and long-term and short-term pattern recognition and memory as well. The thing makes it very complicated is that an excitatory neuron can have very different function or roles depending on where it is and what their neighbors are. So for hippocampus, this is a dissection of the whole hippocampus from anterior to posterior. And if we take a look at the mid-hippocampus section, we could appreciate that there are multiple layers of the hippocampus. The pink one here represents dentine gyrus. So these are layers of neurons or granular neurons. They're small, densely packed, very round. And those are the commanding center in the hippocampus, are making the long-term and short-term memory and also giving decisions to different other regions of the brain. And the CA region, CA4, CA3, CA2, CA1, those are different inhibitory or excitatory neurons that are receiving information from the dentine gyrus, and then they will connect and project to different regions of the brain for signal relay. A very unique circuitry in hippocampus is called the trisynaptic circuitry, where the information is coming from the entorhinal cortex here in purple, and then goes to the dentine gyrus in pink here as the commanding center. And then it goes out to CA3 and goes out to CA1. And CA1 will connect to the subiculum here, and then to the rest of the other regions of the brain, like cortex or midbrain. So it really depends on where the excitatory neuron are. Their function can either be making the decision, making the memory, making the cellular memory, or like being a messenger relay in specific decisions. I know a lot of people are working on single cell and bulk RNA-seq analysis, but one critical challenge in this type of RNA analysis is there's no spatial information. As you could see, the very first step when you are collecting tissue for single cell or bulk RNA-seq is that you need to dissociate the tissue and collect the RNA. And during that tissue procurement, you're either using mechanic or enzymatic dissociation of the tissue to put them into single cell. And depending on the tissue, you might need to do enrichment with flow or with certain marker to get the cells. Because for cell types that under 1% in enrichment in your sample, very likely you will not see them in your single cell study. Every experiment usually can test about 5 to 10,000 single cells in one sample. So if a cell type is less than 1% in your sample, then very easily you will lose those population. And another critical problem is that for neurons, like big cells that contain processes like dendrites or axons, majority of the mRNAs are not in their nuclei or in the regions close to their nuclei in their cell body. They're actually sending out like millimeters away from the nuclear body. So those subcellular gene expression in the cytoplasma will be missed during the dissociation process. And also you will lose the cell morphology when you dissociate the cell-cell contact. And as I mentioned before, like the spatial dependencies between different cells and how cells organize into tissues are also missed. So spatial omics seems to be a very straightforward downstream from single cell, but there are still two big challenges in spatial omics. First one is how we can barcode the tissue when you have a very small number of molecules can be detected. How do you put a location index at the cellular resolution? And the second challenge is how do you capture high enough number of unique molecules at the picogram level? Imagine one single cell can only have 2 picogram of RNAs, but when you do bulk RNA-seq, you already have 10 nanogram. So that would be a 5,000 difference when you are amplifying the library. So technically, you might introduce noise or you might miss the critical RNAs in your library very easily when you are scaling down to 2 picogram. So these two challenges has been previously addressed by single-cell RNA-seq with different barcoding or different detecting technologies. In history, there are two big mainstream. One is imaging-based, one is sequencing-based. So imaging-based methods are using pre-designed probes, such as RNAscope or Murphy-scope or Xenium-scope, Padlock-scope probes that are complementary binding to specific mRNAs. It's very high resolution, but you see what you want to see, and usually it's kept around under 1,000 transcript in total. Another mainstream, it will be sequencing based. Say, I don't know anything about the tissue, but I know all the eukaryotes have the poly A tail in their mRNA. So why don't I just go and capture all those mRNA, and then during sequencing alignment and then analysis, I could just align it back to the reference genome and know what type of RNA they are. So this one is more unbiased and require much less knowledge about the tissue for sequencing-based method. But the downside of it is that usually the resolution is limited around 10 micron to 50 micron. And then there will always be gap between the two capturing location. So having all those technology in mind, we still want to see, is spatial RNA-seq the best method if we want to know what is the Google map of the brain? Can we know who's who at the single cell resolution for their landscape? If we only look at imaging-based method, we can probably see what we want to see for brain, but we may miss out the probes that we didn't design or like validated. For imaging-based method, 55 micron usually contain anywhere from 2 to 50 cells in human or mouse. So we lose the single cell resolution. And one critical problem of that is that those are just defining cell types based on their RNA. But what biology is defining cell types is actually based on their protein, like CD cell surface marker, or their functional marker, or specific marker that are unique to their cell types in flow or in imaging. So there are a big gap between RNA and protein. That whatever you see in RNA might not like be relevant in protein. They may not even be correlated to each other. So how can we address that gap with spatial morphology and also cell identity? There we use a method called spatial proteomics. So this is a method where we're using a multiplex antibody, primary antibody that conjugated with DNA barcodes. So each of the colors here are representing one specific primary antibody. And Dr. Gary Nolan's lab from Stanford University designed a special chemistry to conjugate a 8 to 12 base pair DNA oligos to the antibody with covalent bonds. So it's very irreversible binding of the barcode and the antibody. And there's no secondary antibody. So in the first day, you incubate like 20 or 40 primary antibody conjugated with the DNA oligos with your tissue. And then the second day, we put the tissue onto the machine. So this machine are with vacuum chambers and microfluidic channel that you can have directed flow with specific wash buffer or reagents you define in those bottles. And then every cycle, we can put the complementary DNA barcode with the fluorophore to detect 3 different antibody at a time. So once they detect, we image the whole image, like we image it by stitching specific area at high resolution, usually 20x or 40x. And then because it's a very weak binding for the DNA oligo, so you can adjust the salt solution, the saline solution for binding and unbinding. So you wash it off, and then you have another 3 different type of fluorophores binding to 3 different targets. So this entire multiplex process usually take about 4 days on the machine. And then the raw data is around anywhere from 200 to 400 gigabytes, depending on how big your tissue is and how many antibody do you end up using. And this technology was first developed in 2018 from the Stanford University, but later got commercialized by Akoya Phenocycler. And you can bring your customer antibody and then design your own barcode. And as long as it doesn't hybridize with the existing barcode and you can validate it, it can be used in conjunction with the already validated antibody. So the difficult part is how can you organize your own antibody panel for specific biology you want to study? Because each antibody might work by itself, but they might not work all at the same time. And the mission is how can you have 20 or 40 antibody all work at the same time. So the current pathological IHC standard is usually up to 4 different antibody because you can only distinguish them by 4 different fluorescence channel. But through this cycling imaging that we can detect multiple samples, multiple antibodies at the same time. So one tissue, but multiple antibodies at the same time. And the tissue I like to circle back we're working with is the human brain. So unlike mouse brain, human brain is very difficult to access. And it's not like tumor or biopsy sample where you can ask for biobank resources or archive biobank resources. Human brain, you need a whole different IRB and the tissue collection and consent process. And the brain degrade very fastly. That means whoever are fitting the brain bank criteria, you need to have to send 2 teams specialized. One is the social and legal team to have the relative decide whether they want to do prolonged life support. And another team is the medical team that we need to make sure this body, once pronounced death, you will have like adequate time to go to the cold storage and get transported to a central facility for autopsy and for toxicology. So those cannot be nationwide. Those are almost always regional. So here in the Michigan Brain Bank is 4 hours drive. So any hospital that outside of the 4 hours drive range is hard to be included in the brain bank because the transport and everything after 24 hours, then all the tissue would degrade, like you will not have RNA, and then a lot of things can happen and you cannot actually predict when a human is pronounced death. So the team has actually have to be on call 24/7. So this is really a whole different level of effort for the tissue collection. So I'm showing here as an example for the fixed brain, which is much easier to work with. So the clinical brain bank here is half of the brain will get fixed and then half of the brain would get fresh frozen without any cutting. So this is showing the fixed brain in the left brain. So you can imagine the brain is, the eye is facing the board. And then this is the very anterior piece of the brain, cutting all the way up to the very back of the brain. And there are only 3 brain slices, coronal pieces that are containing human hippocampus. So I'm outlining here the human hippocampus. And the mid region of the hippocampus is marked by the lateral geniculate. So there's a dark region of the nuclei in the thalamus where the visual neurons are crossing at that place. So you will see a like dark region, like a landmark in the mid-hippocampus region. That's how we define. And there are neuroanatomy of human hippocampus atlas that we can refer to where exactly we're getting that and what are the associated neuroanatomy for other like fovea or ventricular space or putamen and basal ganglia. And the beauty of hippocampus is that it's a highly organized tissue. So as I just said, there are dendrite, the commanding center, it has 3 layers. The mid purple layer is the neuron containing dense layer. And then the yellow one are either axon enriched region or dendrites enriched region. And then there's another conoammonous region, like a horn. So there are, say, 4 at the very center connecting to the dendrite gyrus, and also say 3 and say 2 and say 1. So this belt is the neuron containing region in conoammonous, but there's also 2 other layers like molecular zone and the striatum oriens, so the polymorphic zone in the conoammonous. So similar to what you see in dendrite gyrus, they're enriched in axons or dendrites, and we'll get back to that. So in this highly organized human brain hippocampus, how can we map single cell morphology, and how can we understand their cell type? So the tissue we're collecting in total are encompassing of 17 human hippocampus. So we cut all at around the mid hippocampus region for 9 neurotypical controls, and then 8 major depressive disorder human patients. All of them are sudden death, meaning cardiac or myocardial infarction, car accidents, gunshots, stab wounds, street fights, or suicide. So fall from heights. So for this type of patients, we exclude the axonal state longer than 6 hours. So they're all sudden deaths. They're not caused by pre-existing conditions. And they have a very short postmortem interval from the time they declare death to the time the brain was collected. So it's 16 plus or minus 10 hours. So clinically, if the brain was not stored in cold storage 4C degree and is more than 48 hours, the tissue cannot be used for RNA or DNA analysis because they are all fragmented and or they just become like a mushy gel that you no longer have the intact tissue anymore. And for people who are interested in the actual process, because we're only working with fresh frozen tissue, this tissue are collected in right and left brain, and then we cut them into 2 centimeters thick in coronal section. So this region is the temporal lobe. And because it's frozen, so it's like a rock. So we usually put dry ice in a very big box underneath, and then we have this diaper, and then put the brain upside and use a dental saw to cut. So remember the slides I showed, there's only 3 coronal sections will contain the human hippocampus. So you have to have very nice neuroanatomy eye when you look at the brain coronal section, because very likely you will miss it when they're unstained. It's very difficult to tell. Very easy to find amygdala, basal ganglia near that region when it's actually temporal lobe, but does not have hippocampus. So after we cut the entire temporal lobe, we don't do any OCT embedding. We just mount it in the cross section, and then we just section and then collect the center, which is the hippocampus here. It's around 1 centimeter by 1 centimeter. And then we perform the spatial proteomics. So what spatial proteomics can do is that it can stain multiple antibodies at the same time. And this is so far is the best method to construct the single cell atlas in the human brain. And we are the very first team in the world who are doing the human hippocampus, because this technology are so new and the antibody are not so well validated. Previously, when the group are inventing this technology, they are studying immune and gut. So there are only about 40 antibodies developed for CD markers and immune marker. But for the brain, we have to validate every antibody by itself and then conjugate the barcodes and then test the panel together. The difficult part is that when you have a cell, the target can be on the cell surface, in the cytoplasma, or in the nuclei. So how strong do you permeabilize your cell? Antibody itself is rigid. It's not a small molecule that can infiltrate everywhere. So you have to think about what cycle goes first, which 3 antibody goes first. Would they crosstalk? Would they compete for the same location? Or would they compete for for the same integrity in certain regions? So those have to be optimized. It takes about 1 year for us to finalize the antibody panel. But the result is great. where I'm showing a video here. This is the raw data is a pile of individual channel images. So the beauty of it is that you can look at different markers by toggling on and off. So for neuron for all the neurons, neurofilament yellow here for all the axons, and red here, PSD 95 for all the dendrites. And it's thousands of images stitched together at 300 nanometers. So you can magnify to see subcellular resolution, and you can scroll around to see specific crop region for specific things you are looking at. So here is the granular neurons, very densely packed, very round. And then we are looking at the vessel here. So they're looking very different. And we have a cross-sectional like center of the big lumen, and also some macrovasculature at the side. So what makes hippocampus really fascinating is that a lot of the neuroanatomy have been known. Now that we have the images and we have a lot of neuropathologists in the team, can we actually give them a single cell annotation based on the images and then look at all of them at the same time? So just by looking at 6 markers here, we can already identify several different cell types. For instance, these are the granular neurons. Those are usually exaggerate neurons in the dentate gyrus. And there's our oligodendrocytes staying with olig-2 in their nuclei. And they're also big, like triangular shaped pyramidal neuron pointing to the dentate gyrus. So these are typical conoammones neurons. And there are also vasculature staying with collagen 4 in their matrix. So very difficult to tell if you have each of the antibodies staying in different slides, then you don't know how they interact or how they distribute it. We have also gold standard, which is nasal staining in the brain, where you can see here, these are the large neurons where you don't have the nuclei, but you have the axon bundles. And this is the nuclei, this is the axon bundle. And these are the oligodendrocytes, very round, very small, and densely packed. And these are the nuclei of the granular neurons. And this is blood vessels. So you see endothelial cells in the almond shaped, like aligned with the vessel wall, and astrocytes with some processes and irregular shape in nasal body staining. And we naively thought it would be a very straightforward process, but to extract cell segmentation from a deck of images are not that straightforward when you have over 82,000 cells in this single image. So we end up having 3 postdocs in the team figure it out in more than 6 months. So we established a new analysis workflow based on multiplex spatial proteomics data. And I want to shout out to Dr. Yuki Tan from Stanford, from Gary Nolan's group for helping us analyzing this, optimizing their method for brain data. So essentially what we do is that we use the DAPI plane as the anchor point, and then we adjust all the image layer because they might not exact the same Z. So we need to find the anchor point in the glass slide to adjust the Z. And then we use the DAPI as the ground truth for cell nuclei, and use cell nuclei as the mask for segmentation. So for instance here, so all of the cells here having a nuclei staining in DAPI, and then we use this seal host as the cell's mask to extract all of the marker expression within that mask. For neuron, it's different as immune cells because you cannot have a clear cell boundary. One neuron can have over 1 million dendrites, and they can project really long, far away from their cell body. So it's impossible to get the outline of the entire neuron out shape based on their cell body. So instead, we take a step back. So why don't we just look at their nuclei so we know there's a neuron there? And then we do the extraction of all of the 25 protein markers. So each row here will represent one cell, and each column here represents the fluorescence intensity for each marker. And we also have the XY location. So this is converting deck of imaging data to tabular data, which are expression matrix of their protein. And this protein data can later be studied together with single cell RNA-seq and for high dimensionality reduction and other things. So as an example here, this is the antibody panel we use and validated. So we use 6 different markers for neuron. mu(n) for all of the neuron cell nuclei, map2 for axon, neurofilament light chain for axon, PSD 95 for dendrites, GET66 and BDNF for either inhibitory or some excitatory neuron. And there are also some immune cells. In the brain, very likely you will not see a lot of immune cells, but in some of the vessel lumen or in the choral plexus, there are some immune surveillance there, so you will see some immune cells and vascular cells with CD31 for endothelial cells or collagen 4 for ECM in the vessel. The beauty of that is that because we have the XY location and also the cell expression matrix, so we can use Scampi to project them into the n-dimensional space and see how individual cells are different or similar to each other. So here, this is the protein projection. This is not an RNA projection. So just having 25 plex is good enough to see the difference between immune cells here and vascular cells here in brain versus neuron, because this cluster is separated out from other cluster, and all of the cells here are having a expression of mu(n). So it's very definitive marker. It's easy to tell. But for astrocyte and microglia, it's more tricky because they share a lot of the markers together. And depending on their state, whether they're homeostatic or reactive, they can have very distinct functions. So for microglia and astrocyte, you could see that many of the markers are shared for AQP4 or for TMEM119, microglia have a much higher expression, but astrocyte will also have some expression level. The beauty of this is that now that not just one sample, you can compare multiple different samples together. So we put them, so each dot here represent one cell, and then we have 16 human hippocampus in together. So you see the clustering are based on their cell type, not based on their batch or background. And we also calculate the cell composition in all of the 16 samples. So this will be a very comprehensive, very unbiased composition counting of different cell types in each of the sample. And it provides a spatial distribution where you can calculate cell-cell distance, how far a single cell is from each other in control or MDD group. If you see microglia are closer to each other in the disease group, that means they're probably aggregated and they're reactive. We can also compare how many pairs of the interaction are detected in the disease group and control group. Say these are at the 7,500 level, so you can look at all of the samples for their cell-cell distance at the same time. And next, we are just at the very top here, we identify what cell types is there. This is spatial biology, so we want to understand how do they organize together, how do they form neighborhood. So the way we're defining neighborhood is how similar the cell composition in a random rolling window. So we define a 200 micron rolling window and then let it randomize to roll in the image. And if the cell composition is similar in any particular location, they're defined as one neighborhood. So this is an example of the human intestine where they have defined all the cell types and they have their XY location. They perform the neighborhood analysis and can identify multiple neighborhood here. So what are the similar cell composition consistently showing up in different sample? And they can also look at what are the communities, how one neighborhood is always likely to co-occur with another neighborhood. So in healthy intestine, you will see like 4 neighborhood, but in Crohn's disease or IBD disease with inflammation in the gut, you will see specific immune neighborhood containing T cells or macrophage. So the idea is that if you're in the wrong neighborhood, if your epithelial cells surrounded by 10 immune cells, you're going in trouble because you have the wrong neighbors. So this is a very nice way understanding their spatial organization. we adopt their algorithm for the brain. So here, the first image on the left here is the single cell annotated by cell types. The purple represent all of the neuron. The blue represent oligodendrocyte. Pink here represent microglia. So like each of those 82,000 cells have their identity and XY location as the input file. And then we run the neighborhood analysis. So each color here represent one specific neighborhood identified by the analysis. And we can compute the abundant enrichment of each cell type in each neighborhood. And this is what our pathologists annotate the same hippocampus. So you could see here, the neighborhood is not 100% perfect, but you could tell the dentate gyrus and the CA region very robustly by their cell composition. So they're not randomly localized. There's a pattern how they like to stay and how they organize. The beauty of it is that if you have no knowledge to annotate every human brain tissue, you can just run a neighborhood analysis. And this is what the machine can detect as conserved neighborhood in the human hippocampus. So all of the yellow region are annotating the pyramidal neuron layer in the cornu ammonis, and all of the pink layer annotated here here are targeting the granular neuron layer in the dentate gyrus. So without knowing anything in the hippocampus, because you are cutting at slightly different level and at different orientation. So this cell composition can tell you what are the conserved layer in the neuroanatomy. And what's more is that this is very early, we haven't published this yet. So, but this is opening up a lot of opportunity when you have disease group or when you have another group to compare, because this data is served as a reference, as a ground truth for what neurotypical human brain looks like. And then it can be used and compared to any other like AD brain, Alzheimer's brain, Parkinson's brain, or a neuroinflammatory response, multiple sclerosis brain. So it can be applied to many other data as a ground truth. And what's more exciting is that we find that since proteins are not independent, they have their internal relationship. And RNA is the same. RNA, like if you look at GFAP and vimentin, either they are very highly correlated in astrocyte or they are not expressed at all in neurons. So we are using a state-of-art method called MaxFuse, where we can project the single cell reference data onto the proteomics data. So this is the very first method that it can integrate single cell RNA data with protein data. If you look at each antibody, say GFAP in RNA and in protein, very likely they are weakly linked. They are not linearly correlated if you look at individual protein or individual cell. But this method is using like, I'm not just looking at GFAP, I'm looking at 10 different protein around GFAP and 10 different RNA around GFAP gene. And then I'm using your neighbors to define the center gene. And I'm using your neighbor cells to define you. And by iterations, it gives the maximum likelihood for what single cell data is likely to appear in your protein data. So single cell RNA-seq data do not have spatial information, and spatial RNA-seq data does not have single cell resolution. But if you integrate single cell RNA with spatial protein, you can get this single cell atlas of what RNA will look like in this atlas. So I felt this is bridging different modality, saying like when you have some RNA data and protein data and you want to know what the RNA is going to present in your protein data, this can be a very nice method to look at. And together we have collected 16 human brains for hippocampus, and we created multiple atlas for RNA, for protein, and also for spatial epigenome. And we're providing this as a resource to the field as part of the Human Brain Initiative project of the NIH. And I do envision that spatial biology is opening up a lot of opportunities in the brain and in other tissues in general as well. I think for the time, I like to advocate for my lab, and we are a new lab in spatial system biology. So the work I just presented are my postdoc work, but moving forward, we'll be focusing on spatial changes in traumatic brain injury. We're using spatial RNA-seq, ATAC-seq, and also proteomics to understand what is stress, what is injury, how does the brain remodel after injury. And my lab is recruiting at all levels. So if you're interested, please shoot me an email and we can start the chat. So with that, I'd like to thank and acknowledge all the hard work from Dr. Yan Xiangden and Dr. Saima for the technical support, and for the cohort colleagues at Columbia Psychiatry and New York State Psychiatric Institute were providing the top quality human brain tissues. And my PhD mentor, Dr. Rongfan, for developing those spatial technology and establishing the collaboration with Dr. Gary Nolan at Stanford. And my postdoc mentor, Dr. Cam Leong, for helping design and directing of the study of this Atlas resource. So I'll end here. Like, I think we are still at the age of the cubism of Picasso's painting for the same guitar player. All the information is there. It's just, can we approach it? Thank you.
Thank you for that talk. I have a question. So in your spatial proteomics data, the neuron labels, actually, there are two types of neurons, glutamatergic and GABAergic. Is it possible to differentiate the two neurons into two types?
Yeah, for neuron subtypes, so the quick answer for our data is that we cannot distinguish them, because for GABAergic, excitatory neuron also have GABAergic neuron when they're immature, and we cannot differentiate which is immature excitatory neuron having GABA or inhibitory neuron having GABA. So we only have 25 antibody. We did not do that, but it's possible if you can increase multiple like SST, LAMP5, proalbumin, and GABA, and there are actually 16 different neuron subtypes. So with protein only, it's a decision of financial if you want to go further. So the current experiment for spatial proteomics costs about $6,500 per sample. So if you want to add additional antibody, the price will just go up. It's the antibody that are very expensive and difficult to validate. But an easier way to look at neuron subtype is to project the single cell data, because single cell you get all of those inhibitory, excitatory profile out at the same time. So as example here, like when we project, see, these are all the inhibitory neuron, MEIS2, proalbumin, SST, VIP. So this, we have their location in the atlas, so where you can take a quick look where they likely are. And we also tried another sample with the control sample where you can project, where you can project all of the subtypes in astrocytes. So the purple is one type of astrocyte, the pink or the red is another subtype. But for inhibitory neuron, it's a mixture of multiple inhibitory neuron. It's very difficult to validate them. Yeah, for experiment, it's very difficult, but for RNA, it's easier.
One extra question is, I mean, it's general biologically. Have you ever observed bilingual neurons releasing glutamate and GABA at the same time?
Glutamate and GABA. I'm not sure if I get the question. Are you saying a neuron expressing both markers?
Yeah, that's right.
We did not. But for hippocampus, there's a specific neuron called basket neuron. So usually a neuron have only one axon and multiple dendrites. But for basket neuron, they're like chandelier cells. So they have multiple axons and multiple dendrites. So those cells can have GABA and excitatory at the same time. So those are known. It's just usually you need to section like 100 micron to get those big cells. Otherwise, if you only have part of the cell, it's difficult to tell where exactly those two are collecting from. But there are very specific neuron types in hippocampus that are not in anywhere else, and they're not very well defined.
Thank you.
Do we have questions from the online audience?
Well, we had one person raise their hand, but the hand has gone down, so I'm not sure if they saw the question. So, I guess I'll say Avery, if you still have a question, if you want to raise, oh, here we go. We do have a question. All right, Avery has a question. So Avery, you should be able to speak.
Hi, yeah, if you can hear me okay.
Yep, we can.
Awesome. Yeah, it was so cool to see all of this. So I remember you mentioning that a lot of interesting stuff is happening kind of away from the nucleus in these far off projections. And that kind of because of the limitation of bounding these cell bodies, it looks like a lot of the kind of neighborhood analysis is happening still around those cell bodies. Is there any way using this technology you can see going forward to look if there's spatial patterning around specific proteins that are associated like more with the projections, like certain behavior that seems to crop up around, yeah, behavior at like the axon or dendrite level? Thanks.
Yeah, that's a wonderful question. So can we look at actual binding partner, the messenger, the receiver, and the circuitry with this spatial proteomics? The quick answer is yes. So there are people designing animal model where they inject the virus and only neurons who are talking to each other will get infected by the virus and they will all label with GFP or IFP with retrovirus. That way you can tell which circuitry they belong to the same like pathway or they connecting to each other for their axons and dendrites. But usually you can only look at one circuitry at the same time, and it's very costly to build those mouse models. So it is possible, but if you want to look at all of the cells at the same time in human brain, it's a little difficult. In mouse, it's possible, but in human, I doubt you can do anything like that. I know a lot of people in brain development research, they are trying to understand how does the brain have one radioglia going to 6 layer of the cortex, and then they're trying to label specific progenitor cells and how they communicate and how they establish the circuitry. So it's possible, but it will be a whole different research area with spatial proteomics.
Thanks so much.
Oh, yeah, I have two questions. First question is, in addition to providing the results, spatialomics to the field. So what kind of specific biological questions that would be or should be answered by spatialomics?
That's a wonderful question. That's exactly what our lab are trying to do right now. So remember the data we are collecting here are depression patients' brain. So psychiatry is something very elusive to study. There's no even objective metrics to evaluate who actually get depression, who doesn't. All of those decisions are based on self-evaluation. But for many other diseases like brain development, neurodegenerative disease, substance use disorder, or neuroimmune disease, there actually no one changes to cause what protein will have a difference or how their spatial distribution will have a difference. So I think if you want to use spatial proteomics, don't go with psychiatric disease, go with the other thing. So my lab is focusing on traumatic brain injury because we thought this is a spatial disease. And I'm showing here, like when you have a blunt injury on one side of the mouse brain, when you do spatial proteomics or spatial RNA-seq, you can actually look at different pathways very nicely, and they're not single cell changes, they are spatial changes. They are very confined, highly organized, and very specific to different brain regions. As I'm showing here, when you have a right hemisphere have the injury, like you have an immune response right after the injury and gliosis, like first responders going there. So these are the spatial patterns, and we can understand with spatial biology. So if you staying like neuroinflammation marker or stress responders protein here, then you will see a difference, how they aggregate, how they talk to each other. So I felt need to ask the right question with the right data set. That's not just Atlas. Atlas is the first step. Then we build upon it.
So, so what kind of information that is that protein, right? How about RNA or small protein or like transcriptomics? So, what protein probably is important to know the position at the subcellular level. But how about for protein activity or its modification? Seems like it's more
Yeah, great question. So we also do RNA, it's just for the sake of time and the focus of this tool, so we didn't go through it. So this is looking at the same human hippocampus with spatial RNA-seq, with DB-seq. So you could see here, this is histology stain, and on the right-hand side, these are spatial RNA-seq. So you can actually look at the key cell markers for how genes are located and what are the top marker genes of each anatomic location, and what would be the difference of the cell type deconvolution in the human hippocampus. This has never been like profiled before, and you can even look at how cells are distributed together. Say if we project a single cell onto the spatial atlas, you can extract each single cell's distribution, and the row would be how similar their distribution pattern is. And from this picture, you can see all of the oligodendrocytes, they like to co-localize, they share the distribution pattern. While the inhibitor neuron, there are so many of them, and they're almost mutually exclusive. Each of them are controlling a certain region. They don't co-localize at all. So this can be used to study cell distribution and organization. And we also do a spatial ATAC-seq. It's a method developed in my PhD lab where you can profile the chromatin accessibility. And here I'm showing the first one, the first column is the RNA data, and the last column is the chromatin accessibility data. So here I'm showing the motif score. So the red means the motif for CRAB-B binding is very open. There are a lot of peaks we can detect from spatial ATAC-seq. If it's blue, it means the chromatin is close here in the dentate gyrus. So this shows you how responsive each brain region are for different TF. So why would force only having memory in the dentate gyrus? You can see here it's because their motifs are open. Those neurons are ready to respond to the stimuli. But if you look at NFIA for neuron, like they're not responsive in the dentate gyrus. So we're actually combining RNA, protein, and also ATAC to provide a comprehensive atlas of the human hippocampus.
Just to piggyback on that, so I'm a clinical psychiatrist, a geriatric psychiatrist. So can't you still use that in terms of like with depression as a model, like where there are cognitive changes, like they even call it like pseudo dementia, because of some of the cognitive changes that occur with severe depression? You know, would that show similar type findings, do you think?
Yes, we look at the RNA dynamics. The chromatin accessibility are not very different. At least with only 16 human brain, we did not see a big one with 9 versus 8. But with RNA velocity, we see a big change in their dynamics for PCLO and glutamate neurotransmitter transporter gene. So the yellow one means major depressive disorder, and the control one means the neurotypical brain. So we do see a big difference. So the MDD brain are more active. They lose control in their synapse plasticity and remodeling. While the control, they're very well confined in the synthesis of their RNA and degradation of the RNA. So these are the most confined conclusion we can have. But if you look at myelin, then they're not different. They're the same. So their fiber and their myelin shells, their Schwann cells are perfectly normal. So this is like, if you don't see any difference in their RNA dynamics, it will look something like this. But if you see a difference, that means you have more unspliced mRNA from a certain disease, while most of the other control, they're like confined with a certain region. There's not a big spectrum of their RNA rate, synthesis and degradation. But for chromatin, I don't know. I found Dr. Eric Neffler and Huda Hill, they're studying the transcription factor and cellular memory, meaning whatever happens to a human brain, environmental stress or early life event, early life event like loss of parents, like no stable home. So those are like environmental stress that will change the chromatin landscape in the human brain. So those would be the changes in the epigenome that people would more likely to have depression or anxiety in their adulthood. So, but for cell memory, it's so hard to profile them with just chromatin accessibility. I felt there need to be more ground truths for the human brain. The most differences we get are not from their depression state, are from their gender and metabolism state and cause of death. So if they're on drug, like substance use, or if they're having prolonged life support or hypoxia, their brain change dramatically and the RNA will change like even bigger. So the differences you're detecting are not psychiatric difference, but actually metabolism difference or gender difference. But it's a wonderful question. I've been always thinking about that. Like what makes the psychiatric patient different from us? How are their cells remembering what happens to them in their chromatin and in their epigenome?
Biomarkers and elucid.
The biomarkers are the results, but what is the control? What is the gene regulation?
Really amazing work. It's so detailed and so precise. I'm curious with this really unique sample from the human tissues, your thoughts on what is sort of uniquely accessible in human versus in a more model system like mouse or rat or even human primate. And how are from the spatial organization level, how are we going to be able to leverage our model systems and then transport them to think about sort of the spatial organization differences between the model systems?
Yeah, wonderful question. So how can we understand the neuroanatomy and evolutionary brain development across species? NIH has a funding project called Brain Initiative. So there are multiple teams. Allen Institute is in charge of the mouse brain, where they already figure out. But for human brain, it's I think it's 400 times larger in a coronal section. So sequencing wise, it's at least 400 times more expensive than a mouse brain, and its brain is 3D. So not just one plane, but three planes. So people estimate it takes at least $8 million just to sequence spatial for one human brain, and it's not even single cell level. So scale wise, it's difficult, and there are dedicated teams. So people are working on different regions. So instead of having the whole brain mapped out, so we are working on hippocampus. There are team working on basal ganglia, team working on substantia nigra, team working on cortex. So right now we're taking like step by step. For non-human primates, there are different non-human primates. I forget the name on top of my head, but the net sustain group from Yale is working on the non-human primates in adult and try to build the paralogs and build the anatomy like conversion from human to non-human primates. And for mouse and rats, so those are pretty much being figured out for single cell and spatial. But there's a neurodevelopment team to project what stage in the mouse brain are correlating with what stage with the human brain. The difficult part is we don't have enough prenatal human brain. There are probably less than 5 prenatal high quality human brain. For those are usually stillborn. So the baby died in the womb for anywhere from 3 months to 8 months. It's very difficult to get those brains. So how can you correlate those? And for teenager brain, it's also very difficult. So here the brain bank do not have any teenager brain. Only less than 3 brain bank in the US will have some teenager brain for sudden death or high-grade sarcoma that their brain are okay, but there's some other like malignant disease. So I felt we don't have enough tissue to work with, and it has a very high quality for the tissue and the dedicated team to prepare the tissue and to annotate the tissue and to work on different omics. It's a 200 research group total effort. It's the next human genome project. Thank you.